I went ahead and did a quick summary of our data. So it was kind of funny. We were going through the Sweeten Creek stuff, and we went through it pretty fast. And then we're like, oh, should we go on and start the Beetree Creek data? And we're like, no, because we want something to be left for the people that go through the Bee Creek data to do. Like, we don't want them to be bored and like be done in like 15 minutes, right? So we paused. And then it turned out that there was a lot of stuff in the Beetree Creek, as the second group folks can attest. And so these are what our data look like. And I made this graph. You'll have to make your own if you want to make a graph like this. But so these are all the different groups down here. And so these are the Trichopteran families, Ephemeropteran families. There are a couple of cases where we only identified things to order because, I don't know, they were missing appendages and we couldn't tell what family they belong to. So that's one thing that we're going to have to think about during our data analyses. Oh man, and these don't look as different as they did on my computer. But what are some of the big differences? Anything jump out at you? Beetree Creek and the white. James, could you hit the lights all the way on this end? I think it's of those three, that one. Yeah, cool. Any differences jump out at you? Beetree Creek versus Sweeten Creek based on our data? Yeah, that's the one where like Sweeten Creek dominated in nematodes. Uh-huh. Yeah, definitely a lot more diversity. And again, it's hard to see. Again, it looked more different on my computer. Different-er. It was more easy to see on my computer. So these pink ones are Sweeten Creek. But yeah, Beetree Creek got a lot of things here that we didn't see. And or Sweeten Creek has a lot of stuff we didn't see. No, Beetree Creek has a lot of stuff we didn't see at Sweeten Creek. Sorry. Was that what you were going to say, Isabel, as well? And so we did see some EPT taxa, some stoneflies, caddisflies, mayflies in Sweeten Creek. So these are the Hydrocychidae. I think Torrin had like half of these. But 70 hydrocychid caddisflies at Beetree Creek. Whereas Sweeten Creek we had, maybe that's one, I'm not sure, maybe two. Same thing with this mayfly family, Ephemeralidae, which some of y'all saw with these gills going down the abdominal segments there. Yeah, almost 30 of those in Beetree Creek. And Sweeten Creek, we had a few. So anyway, I think it was like I think we were surprised by as many as we saw at Sweeten Creek. I kind of felt like, but then we're even more surprised by how many we got at Beetree Creek. So it's crazy. It's crazy. All right, awesome. Yeah, so those are taxa that are fairly intolerant, right? So like a couple of our papers that looked at invertebrate indices of biotic integrity, caddisflies, mayflies, stoneflies are things that don't do well when the habitats are polluted. So you can break it down by order that way. You can also go down to the family level because it just happens that this family of caddisflies, they're actually net-making caddisflies. And so they build these nets and they hang out next to the net. And this net made out of silk captures organic matter. And then the caddisfly will go and clean that off and eat it. And they actually do okay when the waters get a little polluted because there's more organic matter for them to catch. So sometimes they do okay. So of the caddisflies you'd see when things get polluted, this is the one you're most likely to see. But yeah, so mayflies, caddisflies, and stoneflies, you see where there's less habitat degradation, where it's cleaner. Yeah, and I think we'll learn that through our data probably next week, hopefully. So I know it's always cooler when you learn things by looking at the data yourself rather than somebody telling you like this is it. So we'll get a good handle on it next week. Cool. Any other questions or thoughts on our data? Our invert data? Nice job to y'all sorting these out, getting them. Had more stuff than I thought we might. Yeah, James. Yeah, so that's a good question. So nematodes are fairly tolerant critters, and they it's interesting that we got more some more oligochaetes at Beachy Creek, but nematodes, you generally find them more in polluted waters, and um, yeah, and if yeah, so there could certainly be a little bit of error here and there on this, like any data, but yeah, the fact that we found 50 of these at Sweeten Creek and none at Beachy Creek suggests that there is a difference there. There might have been one or two that were missed. That's always possible. But I think on the whole, we'd expect, our accuracy to be ballpark similar between the two, so yeah. So anyway. All right, cool. So last time we talked about kind of macroinvertebrates in general, how they adapt to streams, a little bit about the different groups. And we also talked a little bit about why conservation is so challenging in freshwater habitats. I'm writing up one of our papers from Nepal, and reviewing the literature, there's just so much evidence that shows that extinction rates in freshwaters are higher than anywhere else. And so I think it's kind of an important thing to keep in mind, because you might ask the question, why is that, right? And so what were some of the points that, these came from a review article actually that David Strayer wrote, that did the mussel tinfoil thing. What were some of the points that he made again, and why that might be the case? Yeah. Mm-hmm. Good, good. We can share some else, Brian? Yeah, but it's a great point, right? Because we know that species area relationships are a thing, right? We know that larger habitats have more species, but we don't. It makes a lot of sense to think about it with like islands in the Caribbean, but not necessarily thinking of rivers and streams as islands. But they sure are, right? They're just aquatic islands in a terrestrial matrix, right? Um, cool. There was this idea mentioned just that they're scarce. If you look at the landscape, there's just a lot less of it. So because of that, population sizes are lower. When population sizes are lower, species are more susceptible to what we call a stochastic disturbance, some random event that comes along and knocks out individuals. This could be in rivers a drought, it could be a flood. When habitats are smaller, you have smaller population sizes, they're more vulnerable to some kind of disturbance. This idea about islands, and then there was one last one that he mentioned as well. What do you recall? Exactly, yeah. So they're at the downhill end of the terrestrial world. So, you know, you know what they say flows downhill, right? Everything, all the bad stuff flows downhill. And that's where you find the river. That's where you find the creek. So in the watershed, you might find terrestrial spots that are not impacted, right? Say we've got a watershed that's 70% farm, right? There are still going to be forested spots there that might be intact. But the water is at the bottom, and so all that stuff is going to get washed into the rivers, and it's inevitably going to find its way to the aquatic community. So because of this, if we have any kind of disturbance, pollution, degradation in the watershed, it's going to end up downhill, and aquatic species are going to be exposed to it. So that's why when we look at the bugs that are in our Cerber sampler at Beachy Creek and Sweeten Creek, we can talk about the whole watershed. We can talk about what's going on. We can talk about the whole, how much of that watershed is impervious surface, how much is forested. Like, we can say that just from looking at a section of this stream right here. Like, we don't have to survey the whole, gosh darn watershed. We look at what's able to live here, and we know stuff's coming in from upstream or not, right? Okay. So I wanted to zoom in a little bit here, and I just want to say, like, it's kind of cool after being here for 12, 13 years now. Like, I can kind of incorporate like my own data in these lectures, which is nice, and like my my students' research, which is cool, because we have these ideas, and it's like, well, do those work here? Have we tested these ideas? Do they make sense? So this Upper French Broad River watershed, as we talked earlier, it flows north. It flows again, through the Appalachians rather than arising out of them. It was flowing before they arose long, long ago. One of the oldest rivers in the world. And so, Dossie's paper for today is going to talk about one of these, about, well, actually, Elijah's paper the other day talked about one of these nationwide sampling efforts, but there have been a couple. And this one, called the NAWQA program, was coordinated by the EPA, and they collected a whole bunch of data. So water chemistry, hydrology, land use, a bunch of stuff. And they did this in 51 what they called study units. And so we're in study unit 46 over here, which is essentially the Tennessee watershed. And then they sampled these in different decades. So the gray ones, the ones where we're situated, were sampled in the mid-90s. This is a massive undertaking, like, nationwide, lots of people working, pretty massive scale, 51 of these study units. So a couple of my research students, this is Dylan Sipkowski up by in Rosman, North Carolina. This is Nicole Dexter, who is one of the largest hog suckers I've ever seen. You see that thing in that net? That's a northern hog sucker. It's ginormous. So they were doing a research project with me a few years ago, and we wanted to see kind of how things have changed, right? Like what's changed since '96, and like what has that meant for the health of the rivers and streams here. I need that thing to go away. Make it go away. It just went away. Okay, maybe it's gonna stay away. Is it just because it was getting pulled on by my thing? The little, maybe that's it. So as part of the NAWQA program, they sampled 15 sites in the Upper French Broad River watershed. And I apologize for the resolution on this. We actually made these figures, but I think I exported it with not awesome resolution or something. But so we're here in the watershed. This is Glens Creek. This is Reed Creek. This is Beaver Dam Creek. So UNC is like right in here. And you know this one. This is Swannanoa River watershed here. In fact, this is the thing, the Bee Tree Creek branch coming in here off the Parkway. Parkway's right here. It straddles the Swannanoa and the Ivy River watersheds. So they sampled a bunch of sites. Swannanoa, Hominy Creek in West Asheville, Cane Creek, Mud Creek comes out of Hendersonville, Mills River in National Forest, Davidson River, again coming out of the National Forest, the headwaters of the French Broad at Rosman, a little river that flows out of, what's that state forest? DuPont State Forest. Then moving downstream, Hominy Creek, creek called Newfound Creek, Reams Creek comes out of Weaverville, Flat Creek comes out of north of Weaverville, Ivy River, here's Little Ivy and Big Ivy, Big Laurel Creek, Sandy Mush Creek, Spring Creek. So they sampled 15 of these sites in the Upper French Broad. And this is what the French Broad watershed looked like in the 90s from a land use perspective, okay? So these stars are each of those 15 sites. Green is the forested land cover. Yellow is ag. And then this pink is developed. And so Asheville's in here. This is the I-40 corridor going across like this. This is I-26 corridor kind of doing this thing. So yeah, Hendersonville down here, Asheville here. Got like Fletcher and that kind of stuff here. So I don't know, what's most of the land use like in this watershed in the 90s? Yeah, forested to a great extent. And the urban development that's happening is more at kind of lower elevations along the French Broad main stem. Okay, so just to give you some numbers, because I like numbers. So land cover in these watersheds. So percent that was developed, percent that was ag, percent deciduous, and then stuff that was least developed, not deciduous, like pine trees, grasslands, and stuff. I don't know, do you guys ever watch Sesame Street when you're young? Do you guys ever hear that song that's like, One of these things is not like the other. You know that song? Yeah, you know it. All right. Yeah, James may have written that song. I don't know. So looking at these different watersheds, I don't know. Any of them stick out at you? Stick out to you for any particular reasons? Mud, yes. The aptly named Mud Creek. What sticks out at you about Mud Creek? And then, yeah, 30% of it developed. Yeah, that's more. Even more than Hominy, which is the watershed. Well, actually, not the watershed I live in. I live in this little dinky Smith Mill Creek watershed, but it's close by. Yeah. So Mud Creek, as well as a lot of ag. So only 30% deciduous forest, which is pretty low. Which watersheds have the most deciduous and least developed forest, or and least developed land? Yeah, Davidson, right? You add them up, we've got 95% deciduous or other least developed. Only 1% ag in the Davidson, 4% developed. You know, you got paved lands and fish hatcheries and stuff. You got a little bit. Any others? Yeah, Spring Creek. That's an interesting one. I don't know if you've ever been there. It flows into the French Broad in the town of Hot Springs. It comes in from the west, and there's a lot of national forest land there as well. A pretty cool place. Yeah, and so Swannanoa, fair amount of developed. But anyway, so all this is to say that like we have differences in land use among all these watersheds. So the question is, like, what does that mean to the health of like streams around here, right? So Brenda Rashley, who's an ecologist with the EPA down in Georgia, decided to analyze these data and see like, take the NAQUA data and see what mattered as far as like the fish assemblages, the fish response. The fish response. What is the fish response? Do the fish care? Of course they do. They just don't tell you. And so it measures a whole bunch of variables, and I'll just kind of go through these. So looked at these variables. What kind of things are these variables measuring? What will this tell us about if we have data on agricultural land cover, population density, urban land cover? Yeah, development in the watershed, right? So if we say something like sort of like human influence like on development in the watershed. Okay. Then also looked at things like drainage area, rainfall, elevation, erodibility. How might we categorize those variables? Yeah, certainly. These are definitely hydrology, right? Or hydrologic, and yeah, it cover. I group them this way. So, sort of like geography, including some of that stuff for sure. So, erodibility of the soil, elevation, rainfall, drainage area. Then these things here, flow, also including hydrology, gradient, sinuosity, temperature. I thought of that as more like kind of what the actual habitat is like in this stream. And then a bunch of these things. What are these green things here? Yeah, water chemistry. So these are not all going to be independent of each other, right? We might expect that as watersheds become developed with more people living in them, more agricultural land, it might change the chemistry, it might change the temperature, right? So these things are all going to be kind of, I don't know, it's going to be kind of a mishmash. It's kind of like, let's just throw these into our analysis and see what sticks. But it's kind of interesting, right? Like how do these differ among watersheds, and then what do it mean for the fish? So we're actually come up with 9 fish response variables. I'm just going to list them. And again, this is an example of something where there's like a take-home message. You don't need to know all these details. But you'll be allowed to take this message home at the end of class. So all these different things. The number of native species in the watershed, number of darter species, sunfish species, sucker species, the percent of the species that were tolerant versus intolerant. You ask yourself, could I look up those data for our particular study? And my answer is yes, indeed you can. this cites a paper by some folks called Saylor and Scott with the Tennessee Valley Association, and they list species and tolerant and intolerant. We can get to that. Percent specialized insectivores, percent omnivores, percent piscivores. In case you're curious, this is what fish is this, you guys? Nice try. Nice try. What might tell us that this isn't a darter, though? Where should we look first? So you look at the dorsal fin. So darter are in, this is a great review, you guys. What order are darters in? No, but they have a similar dorsal fin. You might, like, when you're thinking about this, you might want to, like, be perched in a place that you could see from down on high. Perciformes, yeah, perciformes, yes, brilliant. So darters are in the order perciformes. They've got a spiny and a soft dorsal, and they've got a slim body, and the family is Percidae. This dude, like, what order and family are you thinking based on this dude's fins? Yeah, probably, so Cypriniformes, because it's got a single soft dorsal, and there's two families in Cypriniformes, and you guys, I'm embarrassed. I forgot, what are they again? What are the families in Cypriniformes? Minnows and suckers. The minnows are known to their friends as Cyprinidae, and the suckers are, it's a longer word. It's harder to memorize. It's a longer word. Catastomidae. Anyway, so the suckers, though, have mouths on the bottom of their heads, and minnows don't. So this is a minnow. Now, anybody want to take a guess at it? We've only seen a couple of these, not that many. Or have we? I might be getting this confused. So, for Isabel's research project, we do field work in some of the same sites and some different sites. And I think we saw this at some of our sites. I think we saw it at Sweet Creek. It has a dark spot on the dorsal fin. So this is a creek chub. Anywho, this is an example of a tolerant species. They characterize a tolerant species. What about this fish? Green, a green sunfish. Good. So again, look at the dorsal first. It's got a spiny in the front, soft in the back. So it's either a perciform. Well, no, it's a perciform. So it's either Percidae or Centrarchidae. Centrarchidae, the sunfishes, have the deep body shape. Percidae, the darters, have the skinny body shape. This is sunfish. Yeah, and good. So y'all nailed it. They've got these blue bands on the gill cover, these white margins on the fins. This is a green sunfish. There's another tolerant species. What about this fish? Scanning for answers. What order are we thinking? So look at the dorsals. Spiny, soft, two dorsals. So order perciforms. Just like this dude, it's got a spiny and soft dorsal, but a lot skinnier body. This is family Percidae. Look at the tail. I think maybe just a couple of y'all saw this last time. I'm not sure, but it has a convex tail here. It's a fan-tailed darter. It's got those bands as convex tails. So these are fish we've seen. This is an example of an intolerant species. So they categorized all these species. All right. So let me ask you, as human impacts increase, which of these would you expect to decrease? Any thoughts? So certainly good. Expect the percentage of your fish that are intolerant species to decrease. Yeah, probably darters, right? Because just like the fantail darter, a lot of darters are fairly intolerant. They need clean gravel with invertebrates to feed on, and that can go away pretty easily when pollution starts to happen. So Rashleigh said, that she would expect all these except for 2 to decrease as human impacts increase. So the number of species, some suckers are actually intolerant. The northern hog sucker is. So in this area, predicted that the only ones that might increase are tolerant species and omnivores. Why might you find more omnivores as disturbance increases? These are fish that can feed on both plants and animals. Why might you find more of them percentage-wise as disturbance increases? Any thoughts? Exactly. Yeah. As the habitat's disturbed, if they have a broad range of things they can feed on, they're more likely to be able to persist and be okay than, for instance, piscivores, which are have specific food needs. It's interesting. I was reading today about why they thought, but it's interesting. So specialized insectivores, like darters and things. So anyway, so the idea was you could look at these and get an idea for which sites are most impacted. Okay, so she went in and she correlated those fish response variables with all those other variables that were going on. Okay, this is a little section I like to call the ecologist toolbox. So correlation is a technique we use to evaluate the strength of relationship between 2 variables. Some of you have probably heard of it before. And it looks something like this. If you were to plot the 2 variables on the x-axis and the y-axis, then you plot a bunch of dots out there. Maybe we've got like age and height. Maybe we've got like age on the x-axis and like height on the y-axis, right? So if you get data on a whole bunch of folks, probably do something like this, but at least up to your maximum, you get a correlation. So the idea is it measures the strength of a relationship. And so if you get a cloud of data like this that doesn't have much going on, your correlation is 0. This is a weak correlation. You start to see some direction in these sort of clouds of points, and the correlations become stronger. And then when the points all line themselves up on a straight line, then you get a real strong correlation. Anyway, it's just a way of measuring when one variable increases, does the other one increase or decrease? If the other one increases, we get a positive correlation. If the other one decreases, we get a negative correlation. So we've got a kind of a pattern from lower left to upper right in a positive correlation, from upper left to lower right in a negative correlation. So by convention, we test a null hypothesis, which is what scientists do. H0, that's another way of saying null hypothesis. What do we mean in general by a null hypothesis? What does that mean? You've probably heard it a lot, but it's one of those things like hard to like pin down with words. Anybody ever try to pin you down with words? It's not good. Yep. Yeah. Yeah. So everything you said is right on. The simplest way I like to say it is that a null hypothesis is a hypothesis of no effect. It means whatever effect you're testing for, there's nothing. Whatever variables you say might be related, there's nothing. So in this case, we might say that a null hypothesis is that the variables aren't correlated, right? If we're testing a correlation, a null hypothesis are not correlated. So we have tables of correlations, and then we look at the number of replicates we have, and we can determine the probability of a given correlation occurring at random. And that generates what's called a p-value. So the p-value is the probability of a correlation occurring at random, basically based on our null hypothesis, right? And so by convention, there's a probability below which we conclude that, in fact, it isn't random. There's something going on. Anybody know what that p-value is? Yeah, 0.05, 5%. So when we get a p-value of 0.05 or less, it tells us there's a less than 5% probability of your correlation occurring at random. And we say it's not random. We reject the idea that they're not correlated, and we say they are. So the p-value is the probability of, in this case, a correlation occurring at random, given the fact that they're not correlated, but just kind of based on random stuff. So anytime you see a statistic presented, it should give a p-value, and if that p-value is less than 0.05, just kind of based on convention, we say that it's significant. We say that we reject the null hypothesis. Any questions on that? I know that's something that we come across a lot, and I think it's good to have like kind of a basic kind of understanding of what those terms mean. Okay. Does a correlation imply a causation? No. That's what you say. But I don't know. Let's let Tyler Vigen tell us, okay? Has anybody seen this website? Good. It's going to blow your mind. So this is basically a website that generates spurious correlations, variables that are correlated, but there's nothing going on. So for instance, you may be interested to know that if we look year to year, U.S. spending on space science and technology is positively correlated with suicides by hanging, strangulation, and suffocation. It's kind of a dark one to start with. How about this one? How about the number of people who drowned by falling into a pool positively correlates with the number of films Nicolas Cage appeared in? You can see that, right? That may be causal. There may be, So, in 2007, Nicolas Cage appeared in almost 4 films. There were 120 drownings. 2004, Nicolas Cage was only in 1 film, and there were only 95 drownings. I was looking at these last night with some members of my family. We were laughing. The number of people who died by becoming tangled in their bed sheets is positively correlated with per capita cheese consumption when you look year to year. It is on the rise. What's going on? I guess people are eating more cheese and they're also becoming tangled in their bed sheets. Sorry, we could look at all these. Divorce rate in Maine positively correlates with per capita consumption of margarine, in case you're curious. And I don't know, we could spend too much time doing this. The age of Miss America positively correlates with murders by steams, hot vapors, and hot objects. These may be causal. I do not know. Anyway, but listen. What's that? Total revenue generated by arcades correlates with computer science doctorates awarded in the U.S. I don't know. Is it something I haven't looked? Per capita consumption of mozzarella cheese correlates with the number of civil engineering doctorates awarded. I don't know. Sorry. Fishing boat deaths and marriage rate in Kentucky. I don't know. The point in all of this, and yes, there is a point, is that correlation tells us if variables are related to one another, but you got to be careful implying causation. Just be careful. That's all I'm saying. And be careful if you're in a year where, I don't know, just if there's a lot of cheese eaten, then just be careful when you're in bed. That's all I have to say. If it's a year where you see a lot of people eating a lot of cheese, just be careful the way you're sleeping. Maybe don't have any bed sheets at all. I don't know. Got to move on these data here. Okay. But all kidding aside, like what's going on with those correlations? Have you guys ever heard the term spurious correlation? Spurious is one of those terms I only hear ever used with regard to correlation. Like, is anything else spurious? Anybody ever hear that word used for anything besides correlations? Anything else that can be spurious besides a correlation? I don't know. Something you had never thought about before. Okay, anyway. So why is there a correlation there? So yes, because a critical variable is missing. These are sometimes called lurking variables, which sounds very scary and kind of reminds you of like a hidden haunted corn maze or something. But Yeah, lurking variables. It's a variable that's there but that you didn't measure. So in this case, the reason that the number of films Nicolas Cage appeared in is correlated with the number of people who drowned by falling into a pool is because there is this actually, this is not a great example. I apologize. But in some others of these, this isn't a great example. In some of these, you see an overall increasing trend, as someone pointed out. So in that case, time is a lurking variable. Like they're both increasing over time. So they're both correlated with each other, not because they're related, but because there's been a trend over time, right? This is actually, I copied the bad example. But I don't know what the lurking variable is here, actually. Yeah, James. Yep. Yeah. Or it's really sad that someone you knew passed away in the same place. You go buy ice cream to make you feel better. Yeah, I don't know. Right. Yeah. So point well taken, right? These lurking variables. Okay, anyway, so when we are trying to talk about a causal relationship, there's a different technique we use. It's called linear regression. All right. So back to the upper French Broad River watershed. So again, as y'all pointed out, some of these watersheds are really different from a land use perspective. So this is Mud Creek, a lot of ag land here, a lot of developed land. This is Mills River over here, almost entirely forested. On a somewhat related note, isn't it cool how you see the same pattern on different scales in the environment, right? So this almost looks like a leaf, and these streams look like the vein pattern, and it all has to do with like fractal, interesting mathematical relationships. Okay, so what happened? What was going on here? So she used a multivariate technique, which I'm not even going to try to explain. But what it does, all you need to know is what it does. What it does is it singles out the important variables, the ones that were strongly correlated, okay? So the strongest pattern was that some of the sites had a high amount of rainfall, whereas others had high agricultural land cover, suspended sediment, nitrates, conductance, and sulfate. Okay, and then there was this trend, right? So this means that trends that had high rainfall were low in all of these things. Things that were high in all of these things had low rainfall. And then she looked at the correlation of these fish-based metrics and found that percent specialized insectivores, darter species, and intolerant species were low in this area. That is, they were in areas that had a lot of rainfall, but that were low in all of these things, if that makes sense. So watersheds that had lower agricultural land cover had more darter species and more intolerant species and more of these specialized insectivores, which includes darters. On the other hand, these more agricultural watersheds were higher over here. They had more omnivores and herbivores. So does that kind of go with what Rashley was thinking and what we were thinking about how fishes might be affected by this? It's a couple nodding heads. And it's interesting. So there's only one herbivorous fish species, by the way. Anybody remember which one it is? It's the one that scrapes the algae off the rocks with its cartilaginous lip. The central stone roller, yeah. So this was doing really well in more agricultural watersheds. Why might that be the case? Any thoughts? Yeah, we've got more nitrates, and so there's going to be more algae growing in the water. So for some species, it's a good thing, right? There's more algae to grow, more to eat. But for these other species, not so much. So that was kind of interesting. But it points out the way that natural and human factors can connect. So she argues that this dominant pattern had to do with agricultural land cover. So does anybody know that anybody here from Brevard or the area, kind of, sort of? This gets a lot of rainfall, ton of rainfall. So the upper parts of the French Broad watershed get lots of rainfall. Down here we get less rainfall. And oh, by the way, where's most of the agriculture? All this yellow. It's further down the watershed, right? So these sites further down had fewer intolerant species, more stone rollers, more omnivores. There was a lot more ag going on, and less rainfall. Whereas up in here, the different thing, more darters, fewer stone rollers, all that kind of stuff, less ag going on. And there's a little bit, one other kind of funny thing about this study, she did the whole analysis with and without Mud Creek. It was so different. And you might see why. A lot of urbanization, a lot of ag. So, yeah, so that's happening. Then the second pattern, I had to look at this for quite a while and read over her paper several times to figure out exactly what's going on here, but let me try and give you the Cliff Notes version. Well, what do you notice about these variables, first of all? Any thoughts on this? What environmental variables were important? And what might be like the trend here? Yeah, so iron and manganese, some water chemistry variables. And again, since these are together, these variables grouped out. So these variables were positively correlated. So more manganese, more iron in the water, in watersheds where there was what? So this one is more urban. So here, yeah, more developed, right? So these were watersheds that were more developed. So what's interesting about this, actually I had to write out this narrative for myself. It's kind of confusing. Okay, so native species come out here. These urban streams had more native species, which was weird. She didn't expect that. Now, urban land cover negatively correlated with forest. So forest is over here. Forest, on the other hand, had more piscivores. So this suggests, so for instance, Davidson, Mills, those areas had more intact food chains, food webs, but more species in the urban areas. Now, what's interesting is you think of the urban areas, right? So it's like Asheville, right? Asheville, Hendersonville, Fletcher, those kinds of areas. But what was curious was that if you look at the whole watershed, at the whole Upper French Broad, the watersheds that had higher elevation were more urban. So like Swannanoa, for instance, was a very urban watershed, but it's relatively high elevation when you look across the whole watershed. So there's kind of this connection. So if we remember, so these urban areas had more species, and those included a fair amount of tolerance. So like native sunfishes, for instance, were here. And there was just some other weird stuff going on that the places that was urban and where there were lots of people had higher stream gradients, had higher elevation, a lot of kind of funky stuff. But what was interesting here was that, urban land cover didn't necessarily lead to a loss of native species, because it looked like those native species could hang in the habitat that was created. That is, they weren't used to like a natural habitat that was like pristine, which is really interesting because it makes me think of like, Swannanoa River and what that used to be like and all sorts of things. Anyway, all right, and then just to kind of show you kind of where to go from there. So then with my research students, we did some GIS work where we looked across the whole watershed. We looked at the riparian zone, so like 100-meter buffers all along all the streams in the watershed. And then we looked in the 1-kilometer upstream riparian zone. And we looked at how land use had changed from 1996 to 2011 in all those areas. And so we did a lot of this, looking at aerial photos, like digitizing streams and stuff. And I just so then we looked and we asked the question, how has land cover changed? And we found a couple things. At the watershed scale, there was an increase in developed, a significant increase in the amount of watershed that had been developed, but it was pretty modest. It was only about 2%. There's about a 2% increase in developed land across the whole Upper French Broad from 96 to 2011. And that corresponded with a decrease in the amount of ag and forest. And those were significant using a one sample t-test, but still pretty moderate, right? Only 2%. Like, does that matter? Then we looked in the riparian zone, and we found that in the riparian zone, similar thing. The amount of developed land had significantly increased, whereas the amount of forested land had significantly decreased. So this is the difference, the percent in 2011 minus a percent in 1996. So you can see in the riparian zone, it was a little bit more, about 3%, but still pretty modest. So there was a significant change, but did that matter for like the biotic integrity scheme? Because like this is the thing that I always wonder about. Like you see these studies, you know, like Rashley's, like Roy's, like the one we'll talk about today, and they show these pretty striking differences between watersheds, right? But like the elephant in the room is like, okay, like that's a history up to now, but like to what extent are we still like exacerbating the problem, right? To what extent are things changing now? So we did this and we saw that, yeah, land use is changing a little bit. And so we went back to these sites. So this is French Broad River at Rosman. This is Big Laurel near Stackhouse, North Carolina. And we went back, sampled the same way, did the same things that Rashley did with the fish data. And this, so we looked at fish metrics that might have correlated with the land use change that we saw. And out of all the ones that she looked at, there were only 2 that were correlated. One was the percent of intolerant species. And what was interesting is that watersheds that that increased in their forested cover here, let me back up. Sorry. So watersheds that increased in the percent tolerant species had an increase in the forested watershed, but also in the riparian ag and the watershed ag. So it looked like ag wasn't such a bad thing for the percent intolerant species. If they were actually increasing, and these variables were correlated, which was sort of challenging. So it looks like they responded a little bit here. And then the other thing was that the percent of piscivore species actually increased with increasing development. So it was kind of puzzling, and we need to tease through these. But I think, let's see. I'm sorry, this is a negative. So yeah, this is not just a dash, it's a negative. Sorry, I made this too late at night. So that with the riparian ag and watershed level ag, as that increased, the percent intolerant species decreased. Yeah, that wasn't making sense to me. That's what it was. So these were variables that were negatively correlated with percent intolerant species. So it looks like there are these 2 metrics that are correlating with change over the last like 15 years, essentially. Percent intolerant species and then percent piscivore species, strangely enough, was increasing with watershed development. And so I think this is just because these are large predators like smallmouth bass and rock bass, and they, I think, were able to hang in the pools and stuff in places like Biltmore in the Swannanoa here. So I don't know. What does this all mean? I'm not exactly sure, but the main thing I have to say is that it's easy to show these differences like across watersheds, but what we'd really like to know is like in real time, how are things changing and do we need to manage it? So I think this percent intolerant species is something that might kind of help us do this. You know, maybe this can kind of help us track change in the Upper French Broad River Basin. The percent piscivores, to be frank, we had so few piscivores that I'm not quite sure that this is super reliable, but the percent of intolerant species in our samples does seem to be negatively responding to agriculture development. So anyway, I just thought it's important to remember that like we've seen changes on the order of like 2% in land cover type from 1996 to 2011. But that when we looked across these different watersheds, we see pretty huge differences, right? So percent developed, we have differences from 31 all the way down to 4. So like 27% differences across watersheds. So I just say that to mention that, use that example to say that it's, we can sample site to site and see how differences that have arisen over the last 50 years lead to changes in biotic integrity. But the real challenge now, I think, is to figure out how do we find those more subtle differences and come up with management strategies that'll maintain integrity moving forward in the face of these sort of smaller scale habitat changes which are happening. Okay, so so speaking of different scales of habitat alteration, I can, yeah, you're up, and let me pull it up because I think you actually emailed it to me, right?